This discussion is very important one. The animal kingdom, the body organization, total animal kingdom from the protozoa to chordata. I am going to discuss about the body organization in animals. In this body organization, I am going to discuss about the cellularity, whether they are belongs to the unicellular organisms or multicellular organisms, or tissue level of organization or organ level of organization, and whether they are diploblastic animals or triploblastic animals. How many germ layers are present during their embryonic development at the time of gastrulation, and what type of body symmetry they are exhibiting. Asymmetry, radial symmetry, bilateral symmetry, pentaradial symmetry or bilateral symmetry. These symmetry especially meant for the balance of body organs, and the coelom, very important topic in this body organization is the coelom development in animals. What type of coelom is developing in the animals? The development of coelom is facilitate the arrangement of internal organs in the body. If the coelom is perfect, more organs are present in the body. If there is no coelom, or if the coelom is underdeveloped, very less organs are present in the body. This is our discussion. We are going to elaborate. And also another important topic is the protostomes and deuterostomes development. The blastophore, what is the future of the blastophore? What type of organs are developing from this blastophore, whether mouth or anus? This is our main discussion. Now we can come to the beginning to discuss about the body organization step by step process. The first one is the unicellular versus multicellular organisms. Unicellular and multicellularity is the main thing first we have to discuss, because at the beginning of the evolution of animals, first the unicellular organisms, protozoans, were evolved from the bacteria approximately 2.5 to 3 billion years ago. But later, from these unicellular organisms, multicellular organisms, sponges-like organisms were evolved on this earth. So first unicellular animals, those are the protozoans, and multicellular animals. What are the multicellular animals? These multicellular animals are started with the actually called as a parazoa, porifera. That is porifera. Multicellular animals are poriferans. That is called the parazoa. Of course, they are multicellular animals. Many cells are present in the body. Many cells are grouped and divide the functions, divide the works, and they can cooperate with each other. They can send the cell to cell signaling, but they are living in a group. But this is also cellular level of organization. Although those are multicellular organisms, but that is also called as the cellular level of organization. But after that, in the cnidarians, the group of cells became a tissue. The tissue can perform a function, not the cells. Tissue can perform a function. But cnidarians were evolved as the platyhelminthes and so on. Platyhelminthes onwards, that can show the organ level of organization. The organs, the body can perform a function. But simply we can say that only the protozoans are single-celled organisms, remaining all are multicellular organisms. Level of organization, if you see, tissue level of organization in the cnidarians, cnidoblasts are there, gonozoids, ductallozoids. Many zoids are present in the body. Those zoids are actually made up of tissues, but not organs. That is called the tissue level of organization. But platyhelminthes onwards, we can find some organs in their body. Of course, the digestive system, hooks or suckers or protonephridia or rennet glands, or some of the organs are developed in these platyhelminthes and nematohelminthes. That's why from this platyhelminthes onwards, we can say that organ level of organization. But whereas in the cnidarians, that can shows only tissue level of organization. Protozoans and poriferans, that is cellular level of organization. Then, if we will go to the annelida, many organs are present in the body. Simply, platyhelminthes, nematohelminthes are showing the organ level of organization, but there are no particular organ system level of organization in the platyhelminthes and nematohelminthes. But now Annelida onwards, many organs are developed in their body, excretory organs, digestive system, and system. There are different parts cooperate with each other, and excretory organs and respiratory organs, circulatory organs. Many organs are developed with great cooperation. That is called the organ system level of organization. We found them in the Annelida onwards. In all Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, even in the Chordata, organ system level of organization is present. Diploblastic and triploblastic animals. This is very fascinating thing to discuss during the embryonic germ layers. After the blastulation, the stage is called the gastrulation. This is blastula. In this blastula, only one type of cells are present at the beginning. Now you may observe three types of cells are there, because this is actually beginning of the gastrulation. In the blastula, the gastrulation is taking place. During the gastrulation, three types of cells are forming. These are one type of cells, ectodermal cells. These are becoming as the endodermal cells, and the mesodermal cells are also developing, and they are protruding into the primary body cavity called the blastocele. This is blastocele is called the primary cavity present in the blastula, but now during the gastrulation, some mesodermal cells are developing towards this blastocele, and they can occupy that cells. Next, what type of coelom is developing in the body? That is called the secondary coelom. We will discuss about them after a few seconds. And this is ectoderm, this is called, this is endoderm, and the mesoderm is forming in between the ectoderm and endoderm. After this gastrulation, all the organs are developed from these ectodermal cells, endodermal cells, and the mesodermal cells. From these ectodermal cells, some cells are differentiated. Here a great thing is happening. What is that means? At the beginning of the blastula, only one type of cells are present, but during the gastrulation, three types of cells are developing in this. From this one type of cells, only the three types of cells are developing. That is called the differentiation. One side, during the embryonic development, normally the mitotic division is taking place. Due to the mitotic division, only one type of cells are forming. But here, along with the mitosis, cell division, cell differentiation is also taking place in the embryo. Due to the cell differentiation, different types of cells are forming. Along with the mitotic division, cell differentiation is also taking place. Due to the cell differentiation, from one type of cells, some cells are differentiated as ectodermal cells, some cells are differentiated as the endodermal cells, and some of the cells are differentiated as the mesodermal cells. After the early gastrulation, we found three types of cells in them. Then after the gastrulation, some cells from this ectoderm can be developed as the brain, nervous system, spinal cord, or central nervous system. Some cells are developed as the skin, some cells are differentiated as the nails, some cells are differentiated as the hair, some cells are like that. Some cells are differentiated as hands from these ectodermal cells. From the endodermal cells, some cells are differentiated and form the digestive tract, esophagus, and intestine, stomach, large intestine, rectum, anus. Some cells are differentiating and forming is the different organs in the digestive tract. Some of the cells from these endodermal cells can be differentiated and form the respiratory and the respiratory organs called the lungs also. From these mesodermal cells, remaining all organs in our body are developed. Muscles are developed from, some cells are differentiated and form the muscle cells, some cells are differentiated and forming the areolar tissue, some cells are differentiated as the heart tissue, heart, some cells are differentiated, kidneys, some cells differentiated and form the reproductive organs. Like that, total body is developed. That is called the organogenesis. After the formation of three germ layers in the embryo, that is called the triploblastic animal. In the triploblastic animals, after the formation of three germ layers, organogenesis takes place. Complete organs are developed after completing the embryonic development. But what is diploblastic animals? These diploblastic animals are only, what are the animals? Cnidarians or coelenterates are the diploblastic animals. During their embryonic development, after the blastula, during the gastrulation, only two germ layers are formed. Those two germ layers are ectoderm and endoderm. There is no mesoderm in those organisms during their embryonic development. That's why the coelenteran is present in between the ectoderm and endoderm. That's why those were named as the coelenterata. These are the triploblastic animals. And the body symmetry, this is very, very important topic in the body organization, because symmetry, normally we listen this word in the mathematics. In the mathematics, isosceles triangle can show the perfect symmetry. If you want to cut the isosceles triangle into exactly two halves, okay, you can take any corner, then you can cut the triangle into exactly two halves. If not, you can take a square that can be cut into any two parts, equal two parts. That is called the symmetry. But in animals body also we can find the symmetry. That symmetry means we can cut the animal into exactly two halves by taking a particular axis. If we take axis, for example, my body is there. In my body, there are, you can see, two hands are there, two legs are there, two eyes, two ears. Like that, many organs are in paired format. If you take this axis, middle axis, as the central axis, if you cut my body like this, what happens? You can get the two mirror images. This is called the symmetry. So why the animal's body is exhibiting the symmetry? Because all the body organs are in paired. Those should be in paired because the body should be in balanced. If one hand is there, one leg is there, one eye is there, that is not balanced. That is not symmetry. That animal cannot live properly. Means the organs in our body should be in paired and balanced. If they have to be in the balanced, those should be in paired. Of course, you may get one question, one heart is there, one stomach is there. Like that, some of the organs are in singular. Many, 90% of the organs are in paired, and those should maintain the balance of the body. That's why the animal is living properly on the particular habitat. Okay, but if you want 100% symmetry in our body, you can go to our embryonic development during the blastula period or gastrulation period, you can find 100% symmetry. But after developing the adult organism, you can find 85 to 95% symmetry you can find in the body. However, the body is showing the symmetry. But how many types of symmetry are present in animal kingdom means? But I discussed about the bilateral symmetry first. The example, my body is taken as an example to prove the bilateral symmetry. If you cut the body like by taking the central axis, you can get exactly two halves in the paired organs. But radial symmetry, for example, radial symmetry means some of the organisms, for example, volvox, is round in shape, circular in shape. You can take any axis, then you can cut the animal into exactly two halves. Any axis you can take, that is called the radial symmetry. Then biradial symmetry, for example, in the sea anemone, you can take the sea, you can see the diagram of sea anemone. The mouth is oval shape. If it is the not circular shape, oval shape. If it is the oval shape, the body can be cut into exactly two halves by taking any two directions only. That is called the biradial symmetry. Once you can see any animal, for example, the best example is the corals. That is called the sea anemone. For example, how is the mouth structure you can see. And the pentaradial symmetry. For example, this one, pentaradial symmetry. The starfish is showing this. You can cut, if you want to cut them into any 5 directions only. Any 5, if you take any 5 directions, you can cut the animal into exactly 2 halves. That is called the pentaradial symmetry. But the amazing thing is the larvae of this starfish, Echinodermata, shows bilateral symmetry. You can cut the animal into exactly 2 halves by taking the central axis. This is bilateral symmetry. This bilateral symmetry of the echinoderm larvae are indicating that, are emphasizing that all the chordates were evolved from the echinoderm larvae. And finally, I am discussing about the asymmetry. This is asymmetry, means no symmetry in the body. For example, can you cut the amoeba into exactly 2 halves? No. Can you cut the sponges into exactly 2 halves? No. Those are called the asymmetry. So these are the different types of symmetries. This sponges shows the asymmetry. You cannot cut the animal into exactly 2 halves. No. This is radial symmetry. For example, some of the cnidarians and few protozoans. This is bilateral symmetry. You can cut the animal into exactly 2 halves. This is pentaradial symmetry, that can shows the 5 directions to cut the animal into exactly 2 halves. Those are symmetry. Once again, I am insisting one point is why the symmetry. If the symmetry is present in the animal, the body organs are in balanced format. Beginning organisms asymmetry, and later they become as the radial symmetry or bilateral symmetry. Later they become as the pentaradial symmetry. But finally platyhelminthes onwards. All the organisms are showing the bilateral symmetry except this pentaradial symmetry. This bilateral symmetry is successful symmetry in animal kingdom that can exhibit the paired organs in the body which are in balanced state. And another important thing, very important thing in the animal kingdom is the coelom development. The coelom development, this is called the blastula. During the embryonic development, the blastula is forming. This blastula is containing a primary coelom, first coelom in the body, that is called the blastocele. Then next what happened, our secondary coelom is formed in our body during the gastrulation period. According to the development of coelom in the body by the mesoderm, acylomates are there. Already we discussed about the acylomata in the platyhelminthes. In the platyhelminthes, once again we can see acylomata. Ectoderm is forming and endoderm is forming, but the mesoderm is also forming during the gastrulation, but this is completely occupied by the mesodermal cells and there is no coelom or body cavity in them. That's why these are called A. A means no, coelom means coelom. A, no coelom in the body, that is occupied by the parenchyma or mesodermal cells. These are platyhelminthes acylomates. Next, pseudocoelomates. Once you can come, this is pseudocoelomate, ectoderm and endoderm. The mesoderm is also forming, but the mesoderm is not split into 2. If the mesoderm split into 2 layers, that is called the true coelom. For example, here you can see this is true coelom. Annelida, arthropoda and mollusca, even in the echinodermata and the chordata also the true coelom is formed, the cygocoelomata and enterocoelomata. But now in this, the ectoderm is forming in true coelomates, the endoderm is forming and the mesoderm is also forming. Thus actually this mesoderm is splitting. This is one layer going and attached to the beneath of the ectodermal cells, and this layer is going and attached to the outer covering of the inner layer. So here the mesoderm is splitting like this. That is called the true coelom. If the body coelom is formed by the splitting of mesoderm, that is actually called as the true coelom or eucoelom. In these two cases, acylomata, the mesoderm is not splitting. Pseudocoelomata, the mesoderm is not splitting. These are acylomates, these are pseudocoelomate, false coelomate. But the true coelomates are Annelida, arthropoda, mollusca, and echinodermata, hemichordata and chordates are called true coelomates. But in these cases, Annelida, arthropoda, mollusca, the cygocoelomates. Where is that? In this eucoelomates, the cygocoelomata. This zygosylometa means the mesoderm split into two layers, that is called the zygosylometa. And whereas in enterocylometa, what are the echinodermata, hemichordata and chordata? The mesoderm developed from the endodermal cells, that is called the enterocylometa. Some of the endodermal cells differentiated and forming as the mesodermal cells, then they split into two, that is called the enterocylometa. And the final important thing which we have to discuss is the protostomes and the deuterostomes. Protostomes, proto means mouth, deutero means anus. This is the gastrulation. During the gastrulation, a first pore is forming here. This is called the blastophore. This what is the fate, what is the future of the blastophore is, in some of the animals, this blastophore can develop as a protostome. Proto means mouth. This blastophore can be developed as the mouth. Annelida, Arthropoda, mollusca are called the protostomes. So the blastophore is developing as the mouth. Then echinodermata, hemichordata and chordata, this blastophore can be developed as the anus. Those are called the deuterostomes. These are protostomes and the deuterostomes.